And here's a cartoon showing that. Here is a blood type A, and if you see blood type A, it has protein A around the outside. It can be made by the homozygous dominant version, or it can be made by the heterozygous version, which has got one dominant A and one recessive O. Same thing with the blood type B, except obviously we're using the B allele. With the AB allele, the AB blood type, we have both A and B alleles in there, making both the A proteins and the B proteins. And then of course with the double recessive we have no proteins. This is blood type O over here, and we have no AB proteins on the outside of the cell. Another mechanism of non-Mendelian inheritance is this process called epistasis. Epistasis can be remembered by many genes, one effect, and it works very well in assorted biochemical pathways. So if we have various biochemical pathways here, it works like this. Here is two reactions that then produce a protein that go on to do something. In this case it's a blue protein that makes a blue flower. That way. Here is the gene. The gene makes the enzyme catalyzes the reaction. Here's another gene, the gene makes the enzyme catalyzes the reaction. If we shut down this first gene, then we stop the reaction right here, and we don't get the phenotype of blue. If we shut down the second gene instead, we stop the reaction at enzyme 2, we still don't get the blue protein in the petal. So either one of these mutations, either the W here or the M, either one of those will produce the same phenotype of not having the blue protein. And so this is known as epistasis. Many genes, 1, 2 in this case, one effect, no blue protein in this case. Having said all that, there are some human traits that do exhibit Mendelian segregation and independent assortment mechanisms. This is albinism. It is a recessive trait, and we have a picture here of somebody with no skin pigment, no mendelian. And if we look at the male, he's heterozygous, the female is heterozygous as well. They produce their alleles in their gametes here. We put those on the punnet square like we've done before and just add those together. So here's big A and big A making homozygous dominant. And this person will make mendelian and have normal skin tone. The same thing with big A and little a, although they are heterozygous, they will have normal skin tone. And the difference here is down where we have little a and little a, where we have the albino and no skin tone, and therefore we have the white coloration. Again, we have a 3:1 ratio, so it's showing a normal Mendelian inheritance pattern. There are about 5,000 or so known single gene traits in humans, and this is just one of them. Now, it's difficult to perform Mendelian experiments on humans. You can't really go crossing humans together just to find a 3:1 ratio. So what we have to do is we have to use existing data and predict the inheritance of certain human traits into the next generation. And we can do this by using something called pedigree charts. Pedigree charts are a graphic representation of various generations, and we list different generations by line. So here's the first generation here, the second generation here. So this second generation are the progeny of the first generation. So there are special symbols to represent these different individuals. The symbols, the square is the male, the female is the circle. If you have a male-female mating, it's a single line between the two. If there are two lines between the two, it means that that mating is between relatives. It's what we call consanguineous, or it means between blood. So this could be a brother-sister mating or cousin mating, somewhere around there. We generally put the birth order from left to right, so this male was the first born, this female was the second born, and this male was the third born from these parents here. These are identical twins, monozygotic twins. Now there's twins that are born from one egg, and here we have identical females. If you have the trait that you're interested in, so if we're looking for a particular trait and that individual has the trait, then you fill in the symbol so that it's darker. If we know they're a heterozygote, then we fill it in halfway. And if they're a carrier, when we come to X-linked traits, which we haven't done yet, but we will, if they're an X-linked trait carrier, we put a little dot in here. And that, of course, can only be females, because males only have one X chromosome. So you only have carriers of X-linked traits in females. Dizygotic twins from two eggs are slightly different. If you see here, we have a line between them, and this is no line between them. So we can use this standard form, we can use these standard symbols to show how a trait is inherited and how family members might be at risk for a particular trait. And this is very useful in something called genetic counseling, where if you have a particular trait, or let's say a relative of yours has a trait, you can come into the genetic counselor and you can talk to them and put together a pedigree chart and work out what percentage risk you are for having that disease, or maybe what your children are for having that particular disease. So here is a simple pedigree chart. We have a father and a mother, 3 children, and this P here is something called a proband. And the proband is the name given to the person that came in to the counsellor or came to seek medical attention first of all. So in this case it was the middle child, the son, that initiated the study, and so they get this arrow saying this is the person who did that, and we can start predicting everything else from that. So here's a pedigree chart, and we can see it consists of 4 generations. So we have great-grandparents, grandparents, parents, and the children, and we have, in this case, 6 children from a particular parent here. If we find here that this brother and this sister, cousin in this case, mated and produced 6 children, we find that this person has passed away, this person was stillborn, passed away again, these were twins, this one has the effect, here are some dizygotic twins over here, and so on. So what our goal here to do is put genotypes to some of these symbols and trace them back. And I'll do a few and leave you to do the rest, and we can finish off in class. But let's say we are looking at a recessive trait, and this person here has that trait. We know that that person must be double recessive. Same thing with this person, and this person, and this person over here. If they've got the trait, and it's a recessive trait, they must be double recessive. Well, if this person is double recessive, they must have got this little a from a parent. And so we know that this mother must have little a in it, and the father must also have little a. But because neither of these parents show the trait, then we know that this must be a heterozygote, because here they will be carriers for the trait, but they don't display it. So we know they must be heterozygotes. If they are heterozygotes, then we know that this big A came from one parent, and we'll put the big A over here, something, and we know that the little a must have come from the other parent, over there. And so it's this case of logic, just plotting back to see who has the trait and who has risk for having the trait in the next generation.